Well, I think there's a lot of work to be done before the robot does my gallbladder, but I'm reassured that there will be something that will go boop, boop, and stop somebody cutting my bile duct, and I think that will be probably coming into use sooner rather than later. And thank you for your wonderful lecture and for bringing all this information to us. Exciting times. I have been so impressed over the years with the quality of the research that is being done in this country and being presented internationally and nationally that I felt it appropriate that we should recognize the winners of the three major surgical meetings we have in this country in the year: the FREYER meeting, the WATERFORD Surgical October meeting, and the SYLVESTER O'Halloran meeting. And Council has agreed to my wish that we would have a presidential medal and that we would have that decided at this meeting with the presentation for all of us of the three winning presentations. And Derek Alderson, Melina Kebby and John Alverdi are going to be giving us a discussion at the end of each of the papers and marking, and their deliberations will decide the winner of the President's Medal. So I would like to ask Dr. Ola Ahmed to come to the podium. There you are. And Ola is going to give us a presentation on expanding the liver donor pool in an area of marginal organ access, the role of antioxidant defense mechanisms. This was done in WashU, I know, but Ola is an Irish graduate and has really, when I heard this paper, it blew me away. So you're very welcome, and I look forward to hearing the paper.
So good afternoon, everyone, and thank you to RCSI and to Professor O'Connell for the opportunity to present on this session. And over the next 7 minutes, I hope to talk about marginal livers and in their role in expanding the donor pool. So recent epidemiological data has shown a 500% increase in the incidence of liver disease over the past 2 decades. And now the greatest challenge facing transplantation worldwide is the shortage of donor organs, placing a huge demand on liver transplant waiting lists. So an important question to ask is, has the donor pool been compromised? And what population or demographic shifts may have impacted this? So this is a study from the University of North Carolina, which assessed current donor characteristics to predict future liver transplantation trends. And the model predicted that we could expect a 44% decrease in donor livers over the next couple of decades. And the reasons for this decline could be related to an increase in obesity, older donors, and underlying metabolic conditions. But ultimately, this could result in 2,000 fewer liver allografts per year. And we can expect to see an increase in the prevalence of marginal liver offers in transplant. So the concern about marginal livers is in their inability to withstand ischemia-reperfusion injuries, which will have a huge influence on patient-reported outcomes. So IRI is a complex process with several targets, but one approach is to minimize oxidative stress or to improve a liver's ability to withstand ischemic injury. And one important antioxidant axis is the NRF2 pathway, and this is a transcription factor that regulates anti-inflammatory, antioxidant, and cytoprotective gene expression. So with this, we hypothesize that NRF2 is impaired in discarded livers. And over the next couple of slides, I'd like to talk about how our studies might fit with current discussions regarding marginal liver use by measuring hepatic NRF2, correlating these levels with clinical performance, observing how machine perfusion may further drive the axis, and finally, discussing potential for therapeutic intervention. So we collected samples from discarded livers that were deemed unsuitable for transplant, and we performed several protein and staining analyses to determine their expression. So one of the first things that we saw was that there was a wide variation in protein expression in our donor group. And we began to see that some livers had a higher expression than others. So we decided to stratify this group into a high NRF2 group and a low NRF2 group. And this really reflected the reality of clinical practice and that there's a lot of heterogeneity in organ discard. So we mimicked a prior study from St. Vincent's Hospital in Dublin. And when we looked at our two groups individually, we saw that livers with lower expression of NRF2 had higher degrees of LFT derangements, they had higher glucose, and they also had less albumin. And one way to look at these is in terms of injury, metabolism, and functional capacity, in that livers with higher expression levels of NRF2 appear to do better in all three parameters. So then at a microscopic level, the high NRF2 group had lower percentages of microsteatosis and lower inflammatory scores. On immunofluorescence staining, we could see that these livers also had less lymphocytic infiltration. Glycogen staining of the livers also showed that low NRF2 livers had higher glycogen content. And when we looked at all of this data together, it could suggest that livers with a higher expression level of NRF2 can demonstrate favorable clinical parameters, despite being rejected for transplantation. And NRF2 expression levels may help us decide which of these livers could be suitable for surgery. So we wondered whether NRF2 expression could influence hepatic responses during normothermic machine preservation. A machine perfusion is really a novel strategy in liver transplantation where the liver is placed onto a machine following retrieval and before implantation. And this is one of the first reports from King's College where 20 livers were successfully transplanted following normothermic preservation. This was followed by a large European randomized trial with over 220 liver transplants comparing this approach to static cold storage, and the authors reported a 50% lower rate of organ discard. So then taking this one step further, the VITAL trial from the University of Birmingham have successfully transplanted 71% of marginal livers that were previously declined for transplant by transplant centres in the UK. So using a lab-based normothermic machine perfusion setup, 8 of the discarded livers underwent 6 hours of perfusion, and we assessed viability in the context of NRF2 using similar VITAL criteria. So we found that following 6 hours of machine perfusion, livers from the high group further increased levels, whereas livers from the low group either appeared static or reduced their protein expression of NRF2. The high NRF2 group also had less injury in terms of LFTs compared to the low group. They were able to maintain glucose levels after perfusion, and most importantly, they demonstrated evidence of effective lactate clearance, which is one of the essential markers of liver viability following machine perfusion. Higher NRF2 expression livers also demonstrated less lymphocytic infiltration, measured once more here in immunofluorescence staining when compared to livers with lower expression of NRF2. So we saw that there was a substantial proportion of our studied allografts that were steatotic. And because of the increasing prevalence of obesity, whereby now 1 in 4 adults seem to be affected, and the associated rise in hepatic steatosis, we wanted to further study this, and I'd like to share some preliminary data. We saw that lean livers had significantly higher NRF2 expression levels, and this is in line with reports of increased oxidative stress in fatty livers, which also had higher transaminases in lean livers prior to recovery. So we then wondered whether the NRF2 axis could play a role in all this, and because our studies to date had indicated that lower NRF2 was consequential, we hoped that increasing levels physiologically could offer another layer of protection to marginal allografts, specifically steatotic livers. So as our final set of experiments, we identified an NRF2 agonist and we designed a fatty liver rodent model, and we treated steatotic rat livers on an animal normothermic machine preservation apparatus. Encouragingly, we saw that NRF2 modulation worked in steatotic livers, and we could restore NRF2 levels in steatotic allografts to lean liver levels. We identified that an NRF2 agonist could also result in decreased arrangements in LFTs. And although this was a small-scale animal study, it can be viewed optimistically as we begin to see more marginal allografts and more offers in transplant. So to conclude, there is a need to expand the current donor pool, and this remains one of the greatest challenges facing liver transplantation. Higher hepatic NRF2 levels appear to result in consistently better clinical and functional parameters. And even in a small-scale study, stratifying grafts using NRF2 expression for machine perfusion could optimize allograft selection, especially in the era of increasing obesity. And NRF2 modulation could be the reality going forward. So such strategies to optimize the use of marginal livers could, whether it be in a small country like Ireland or globally, produce a small but non-negligible increase in the number of transplantable organs and help expand the current donor pool. Thank you to everyone who has contributed to this work, and most importantly, thank you to the donors and their families for their gift of life.
Wow. Panel. Can I ask John Alverdi? You are writing furiously, John.
Oh, yes. Thank you. I was sending a note to my wife. Sorry. No, I listened to the whole thing. It was wonderful. Congratulations. Beautiful, elegant methods and work. My questions are the following. The human data appeared highly probabilistic, not deterministic. In other words, you had a lot of between-group comparisons with variability within the groups, and you didn't really reconcile the within-group variability, which is why you moved to the mouse model, which is to prove that this, in a rigorous way, that this pathway, NRF2, could rescue these livers and make them more palatable. I asked this question, and you'll see it in my talk if you stay for it, which is, were these mice diabetic, fed a high-fat diet? Were they have fatty liver infiltration? Were they like the patients that you plan on modulating the livers on?
Thank you very much for that question, Professor Alberti. So yes, as you mentioned, in our human group, we saw that there was a lot of variability. And again, that kind of reflected the reality of clinical practice. A lot of high-risk livers are high risk for various reasons, whether they're from older donors on top of being steatotic. So that's why we wanted to assess this in a controlled setting in the animal model. So our animal experiments were all standardized. So we had a 60% high-fat diet for a total of 8 weeks. And then we were able to assess hepatic steatosis and H&E staining. And so the average steatosis in our animals was between 30 to 35%, which would be consistent with steatosis in the realm of liver transplantation. They weren't diabetic, but we specifically wanted to focus on steatosis because we felt that in our human studies, 45% of the livers were discarded because of steatosis, and that was the main reason. And then going forward, we hope to look at maybe DCDs and older animal models as well.
Thank you.
Wonderful talk. I have a similar question related to what was just asked. I'll limit mine to just 2 questions. One, why NRF2? There are so many antioxidants that you could be looking at. I'm just curious as to why your group decided to look at NRF2. And then about the variability. On the livers, I'm very curious, given the variability, how many samples did you take in each human liver throughout the liver? Because I would imagine 1 inch away or 1 centimeter away, NRF2 could be wildly different. And so in order to really use this as a predictive model going forward, I would think you would have to have many samples, almost like when we're doing prostate biopsies, many samples throughout the liver lobe.
Thank you very much, Professor Kippi, for that question. The reason we opted for NRF2 was, first of all, this study was done maybe over 20 years ago in St. Vincent's Hospital. And we found, based on that, we wanted to build on that. And NRF2 is an important antioxidant pathway. The reason being that it seems to affect multiple other pathways. It's involved in metabolic regulation. It's involved in anti-inflammatory and cytoprotective gene expression. So that's why we wanted to capture, I guess, a master gene expression. We also wanted to study it because it's a model. It's a pathway that can be manipulated, and it's certainly manipulated in other non-hepatic clinical settings. So in neurological conditions such as stroke, in retinal ischemia, in diabetic nephropathy, it is an antioxidant that is very interesting in other non-hepatic settings and has been mimicked in the hepatic centers. So we wanted to pursue that further. The other question about the variability, so when it came to the serum samples, we took perfusate. So that could give us a global view of liver function tests and other biochemical analyses. When it came to the immunofluorescence staining and the H&E staining, those biopsies were taken from 4 parts of the liver. And then when we were analyzing this, we did 4 quadrant analyses of the actual sample to make sure that we've gotten the whole view of the liver.
Thank you.
Question.
Very briefly, so you're suggesting this could be a useful marker, but you didn't tell us about the time course of when you can get the result in relation to the perfusion process. So just tell us about the fact that this is practical, useful, and could be employed in clinical practice.
Yes. Thank you very much, Professor Alderson. So for these experimental settings, we did protein analysis. We did things like Western blotting, we did immunofluorescence, which can take a day or two to actually get results. However, one thing that we're currently working on is looking to see if we can do ELISA testing, particularly on the perfusate, because I think that would be useful and it would have a high turnaround. I'd also mention that one of my colleagues in the lab here, in Professor Farrelly's lab in Trinity, is also looking into transcriptomic analysis to see if there is one specific biomarker that could be indicative of an intact NRF2 axis, and that would be certainly much more useful in a clinical setting.
Okay.
Well done, and thank you very much, and thank you to our discussants. Our next speaker is Niall Hardy. Niall? And Niall is going to talk about clinical application of AI for characterization of rectal and liver lesions. No pressure, Niall, following on the Johnson & Johnson lecture, so you're very welcome.
Thank you very much, Professor. As you said, my name is Niall. I'm a surgical trainee, and I'm really delighted to be here today as the William O'Keefe Prize winner to present some of my research work. So surgery is about seeing tissue in front of you, making a decision, and then carrying out an act based on your best judgment. My work has been about using fluorescence guidance in combination with artificial intelligence to identify cancer with high levels of certainty within moments. When it comes to liver surgery, it's becoming increasingly common for colorectal metastases, and a traditional open surgery like you see here, there's been a very heavy reliance on tactile feedback given the uncertainty of where even the most superficial of liver lesions can exist. But in an increasingly minimally invasive world with a vogue for non-anatomical parenchymal preserving resections, how do we overcome this lack of tactile feedback? And especially important in hepatobiliary surgery, where it lags behind its other specialties when it comes to the percentage of operations performed in a minimally invasive fashion. Now, ICG was hoped to have fixed this problem, and these slides kind of make you think it is solved. The dye is given here a number of hours or usually days in advance. The surgeon then comes and interrogates with an infrared camera, and you hope that it's trapped where it should be and that it's not in healthy tissue. But this approach really has largely failed for a number of reasons, mainly due to poor sensitivity, poor specificity, as well as logistical issues with regards to giving this to patients hours or days usually in advance. So our goal was to characterize colorectal lesions using real-time dynamic administration of ICG through the tissues. So this is an example of a dynamic process. This is a common use of ICG. This is a colorectal resection. Here the vasculature has been ligated. The dye is given by the anaesthetist. It's starting to flow in here, and it's either shown by presence by green or white, depending on which screen you're looking at. And this is the dye bound to albumin traveling around the circulation. Now, our group has quite a large body of work at this stage looking at characterization of significant rectal polyps using this dynamic approach. And we call this a biophysics approach, an understanding of the biology of the tissue and the physics of the dye through the tissue. And going into the liver work, we had kind of strong confidence that this would work. I looked at 65 significant rectal lesions and got an area under the curve, or area under the receiver operating curve, of 0.93 for these 65 lesions. Now, just for reference, 1 is a perfect score, perfect sensitivity, and greater than 0.9 is an excellent score. So we believe in this process. We believe that it's discriminatory, and for the first time, we applied it to liver lesions. So what does it look like? Here I've got a very obliging SHO to lean the camera on. It just looks like a standard laparoscopic camera. This is open surgery. At laparoscopic surgery, this is the kind of view you get. This is a white light image in the main screen and the top left, a black and white mode on the left in the middle image, and a green overlay on the bottom. For simplicity, I've just removed 2 of those pictures. You've got a white light on the left and an NIR mode on the right. Here the anaesthetist has given the dye intravenously at the top of the table. It started to wash through the entire vasculature. Everywhere is perfused, so it all goes white. But then you see over a matter of seconds, these 2 tissue regions start to discriminate out. And this is based on the understanding of how ICG behaves through healthy liver tissue, which is the excretory organ of ICG, so it's accumulating, and it doesn't do that in the colorectal met. It's not an excretory organ, and therefore it's starting to wash out at a different pace than it was in a healthy liver. So we can kind of look at that subjectively, but how do we objectify that and how do we start quantifying it? Well, here I'm picking two regions of interest. Region 0 is in the cancer, and region 1 is in a control healthy region. Using a bespoke tracker, this is just my laptop screen. I'm drawing boxes where I suggested there in the healthy, in the healthy region and the unhealthy region. And what we see here is the inflow is quite well matched. We saw that visually. The two lines are climbing beside each other. But then very quickly there's a divergence. And this was based on what we saw earlier on, that the behavior of the dye through the tissue depends on the tissue type. And this is happening, as I said, within moments. And this is the kind of prediction. This is, you get whether it thinks it's cancer or healthy and a percentage confidence. And I'll go through how we do that in a second. With regards to results, we did this for 10 lesions, 9 colorectal liver mets, 1 benign liver cyst. And with this cohort, we're getting perfect sensitivity and specificity using this method. So what exactly do we do? Well, this is a sample curve here. This is a time fluorescence curve. And what I want to do is start extracting features from these curves. These are things like the time that it takes for the fluorescence to peak, the upslope of that climb, and then the downslope at different time points. And the point of all this is explainability and interpretability. We can describe exactly what we're doing, and that kind of makes sense from our biological understanding of the dye and the tissue. You can look at these different time points manually as well. This is me doing it on Excel. And the point here is really to show you that there is strong significance between these different time points when you compare unhealthy and healthy tissue. Building the case for explainability and interpretability, we can look at it under the microscope. Here you can see on the H&E on the left-hand side and a fluorescence image on the right-hand side. The dye is within the liver parenchyma and it's no longer in the colorectal met. What we're seeing macroscopically with the camera is also what's happening microscopically. Now, we don't really look in surgery at regions of interest like this. We look at the whole screen. What might that look like? Well, here we're tracking regions of interest, stabilizing the image, and then going to redisplay it with a curve for every single pixel on the screen. So these are just 3 sample pixels, but we can reproduce images based on the dynamic pattern through the last few minutes. So back to the original problem, if you can't touch a lesion, can you start to demonstrate it? If you think there's a hidden lesion, we can see this one on the left here, but there maybe is more. We look at the 2D reconstruction and you can see here the extent of this second lesion that's hidden behind, underneath the parenchyma. Benign lesions, we said they're different, the patterns are different. And similarly on 2D imaging, they don't show any difference here because the perfusion signals are the exact same. This is clustering. We did this in the operating room. This is showing, this is an unsupervised machine learning method where it asks the computer to pick 2 groups. And here you can see the left-hand side, the purple group, the right-hand side, and the green based on their perfusion pattern. And you can start to see the beginnings of a cut line here for operative use. So in conclusion, for the first time, we've characterized colorectal liver metastases by their dynamic perfusion patterns using easily interpretable features, but only easy by machines. This is important clinical uses as we move to minimally invasive surgery and future directions. We now have a pipeline and a protocol for international recruitment. We want to open source this and constantly improve the algorithm as we get more data. Thank you very much.
Well, Niall, that's fantastic and beautifully presented. Thank you. Melina, can I ask you to lead out?
Yeah, simple question. So wonderful work. So how many subjects so far have you evaluated in developing this model?
Yeah, so this kind of is a basic science approach. Does ICG tell you whether something is cancer or not? And we've looked at over 80 significant rectal lesions with very high results there. We've looked at 15 liver lesions now. So we're encroaching in on about 100 patients with in excess of 90% area under the curve for those
What's your
patients.
goal before you're going to publish this work?
We can talk offline maybe.
Oh,
Where are you going to publish it?
I hope you've calculated what the number needs to be in advance.
Yes. So the next step of this is an EU Horizon grant, which has been awarded. It's a 4-year, €5 million project, and we've got 7 centres to run out this thing to increase numbers.
Good answer. Derek.
I may have missed it, but just tell us about the depth of penetration, as it were, of ICG in the human liver.
Yes, very good question. So depth remains one of the issues for deeper lesions. Traditionally, it has been quoted about 1 to 1.5 centimetres of penetration for ICG. Now, there are more camera systems coming online as this is becoming more popular that are giving more depth. And that's going to get better or to reassess as you go deeper. But certainly it is a limitation of this for surface.
Just ask you a supplementary to that then. If we look at people who have subtle, small, non-white light visible surface lesions, how many of those patients harbour deeper lesions within their liver that similarly have not been identified by a preoperative method?
Yeah, that's a very good question, and I don't think this approach is going to be something that's going to solve that kind of problem with regards to ghost lesions or smaller lesions. Certainly, a very significant percentage of people. Yeah.
Okay. Thanks.
Yes, thank you very much. Great presentation, nice job, exciting work. I realize this is work in progress. So my question is along the lines of Professor Alderson's, which is, how the problem I have with all this machine learning or AI is, how does this discriminate in lesions in circumstances of uncertainty, just as Professor Alderson just mentioned? It's one thing to say, look, the lesions you're showing us, they look like any of us could say that looks like a cancer, we biopsy it. But the trick is, for AI, including in the common bile duct circumstance, is that you have an electronic jury or some type of jury that says, I don't know if that's cancer or not, but then that machine says it is or isn't, and it's 100% accurate. And so it's the circumstances of uncertainty. And this was done, Marco, with Barrett's esophagus, where people would take pictures of the esophagus by endoscopy and send it out to an electronic jury and say, is that short, long segments, deep, bad, dysplasia or not? And then the machine can tell you whether the jury is right based on the inter-rater variability and based on the accuracy of these circumstances of uncertainty. Can you address that?
Yeah, I agree. I think with regards to work ongoing, it's work ongoing to become widely used. I think the principle that this technology is discriminatory, I think, has been pretty well proven with those series of patients now at this stage.
Right, in obvious lesions.
No, I don't think so. I think when you look at...
Well, you have to show that.
For sure, yeah. So when you look at, say, for endoluminal lesions in the colorectal setting, we are not good at all as operators at telling cancer from not cancer. We know that very well. There are some very obvious lesions, for sure. We also know that biopsy in about 20% of cases or more in significant polyps will also be wrong. Now, with the results that we're getting with this kind of approach, being able to assess the whole lesion rather than taking representative, because heterogeneity is a hallmark of cancer, we're able to assess the whole lesion using this technology, a kind of an optical biopsy. And in the cohort of, as I said, 65 patients, we are getting an almost perfect score for all of these lesions, including ones that are biopsied in the clinic and have often been re-biopsied. And when we look back at this, the AI could tell us earlier on because of the area that it can map.
Niall, thank you so much, and congratulations on your work. And now we're greeting Maria Davern. I think, Maria, you're online. Are you here? No, you're online. There you are. So you're very welcome. And Maria is going to tell us about cooperation between chemotherapy and immunotherapy to enhance anti-tumor T-cell mediated immunity in esophageal adenocarcinoma, the implications for synergistic combination regimens. Maria.
So I would like to begin by thanking the organisers for the opportunity to speak during this session. So standard cancer therapies are emerging as a valuable tool to convert immunologically cold tumours into hot tumours. And cold tumours are typically characterised by a lack of infiltrating T cells and a lack of a pre-existing anti-tumour immune response, and typically carry a poor prognosis, whereas hot tumours are characterised by tumour infiltrating T cells and carry a better prognosis, whereby the addition of immune checkpoint inhibitors, which block inhibitory immune checkpoints, can reinvigorate exhausted anti-tumour immunity, whereby they can synergize with chemotherapy or radiation to improve clinical outcomes for OAC patients. However, a significant clinical challenge is that cold tumours typically do not benefit from immune checkpoint inhibitors because they lack a pre-existing immune response to reinvigorate. So the standard of care for esophageal adenocarcinoma patients includes perioperative FLOT chemotherapy regimen or neoadjuvant cross-chemo radiation treatment. And there is emerging interest in combining standards of care with immune checkpoint inhibitors in the neoadjuvant or adjuvant setting to boost clinical response rates for patients. So the aims and methods of our study are as follows. Firstly, we aimed to profile immune checkpoint expression on T cells in OAC patients and determine how this might correlate with clinical features and clinical outcomes. So in collaboration with St. James's Hospital in Dublin, we received treatment-naive and post-treatment blood and tumour biopsies from OAC patients, whereby we immunophenotyped circulating and tumour infiltrating T cells for immune checkpoints and T cell activation markers, and then correlated these findings with clinical characteristics, with the clinical potential here of identifying novel targets for OAC patients. The second aim of our study was to determine if first-line chemotherapy regimens are capable of warming up the tumour microenvironment in OAC patients. So we isolated PBMCs from treatment-naive OAC patients, and we treated them ex vivo with a cocktail of either FLOT or cross-chemotherapy regimens. And then we carried out immunophenotyping of T cells, whereby we determined if these regimens could enhance or dampen T cell activation status. We also assessed if the post-FLOT or post-cross chemotherapy tumour cell secretum could enhance lymphocyte-mediated killing of OAC cells. And the clinical potential here was to identify immunostimulatory chemotherapy regimen to combine with immune checkpoint inhibitors with the goal of broadening the efficacy of immune checkpoint inhibitors to a greater spectrum of patients. So firstly, we profiled immune checkpoint expression on T cells, both in circulation and infiltrating tumour tissue in OAC patients, and its association with pathological response rates. So here's a list of the immune checkpoints that we identified on T cells in OAC patients, and those that are highlighted in red were significantly upregulated on T cells in the tumour compared with those in circulation. Interestingly, we also found that increased CTLA-4 expression on circulating T cells positively correlated with tumour regression rate, indicative of a poor response to treatment. In addition, increased CTLA-4 expression on tumour infiltrating T cells positively correlated with nodal metastasis and lymphovascular invasion. So next, we looked at the effect of FLOT and cross chemotherapy regimens on T cell activation status within the tumour microenvironment. And we found that CD69, which is an important marker for T cell proliferation and activation, was significantly upregulated on tumour infiltrating T cells post-cross chemoradiation treatment. However, we found that CD27, which is another marker of T cell co-stimulation and proliferation, was significantly downregulated post-FLOT and cross chemoradiation treatment, which is an unfavorable effect. However, we found that when we added in immune checkpoint inhibitors at the same time as FLOT chemotherapy treatment ex vivo, we could attenuate this downregulation of CD27. So overall, we found that FLOT and cross chemotherapy treatment altered T cell activation treatment via upregulating CD69 and downregulating CD27, the effects of which could be attenuated with the use of immune checkpoint blockade. So finally, we asked or investigated if the effect of FLOT and cross chemotherapy tumour cell secretum could enhance lymphocyte-mediated killing of OAC tumour cells. So very briefly, this is how we generated the post-chemotherapy tumour cell secretum. We treated OE33 OAC cells with either FLOT or cross chemotherapy treatment for 2 days. We then washed off the chemotherapy treatment and allowed the tumour cells to grow for a further 48 hours, after which we harvested the post-chemotherapy tumor cells accretum. In parallel, we isolated PBMCs from treatment-naive OAC patients, and then we expanded these cells ex vivo in the absence or presence of single-agent nivolumab, ipilimumab, or dual immune checkpoint blockade, whereby we then co-cultured these expanded immune cells with tumor cells in the absence or presence of post-chemotherapy tumor cell secretum. And you can see that we found that dual immune checkpoint blockade achieved a greater level of tumor cell killing compared with either single immune checkpoint blockade alone. In addition, we found that in the presence of the post-FLOT tumor cell secretum, the efficacy of dual immune checkpoint blockade was propagated. So in summary, we identified a range of immune checkpoints expressed on T cells in OAC patients, which correlated with poor clinical outcomes, identifying a range of new potential targets in the neoadjuvant setting for OAC patients. We also identified that the chemotherapy drugs used in the FLOT and cross chemotherapy regimens could firstly enhance anti-tumor immunity via upregulating T cell activation markers and enhance lymphocyte-mediated killing of OAC cells. However, we also found that these drugs could suppress anti-tumor immunity via downregulating the co-stimulatory marker of CD27. However, the addition of immune checkpoint blockers could attenuate this downregulation. So in conclusion, we demonstrated that standard chemotherapy regimens used in OAC could potentially warm up the tumor microenvironment, thus highlighting potential synergy with immune checkpoint inhibitors in treating cold OAC tumors in the neoadjuvant, but also in the adjuvant setting to potentially boost clinical outcomes for OAC patients. And future work will include identifying in ex vivo and translational clinical studies what are the best immune checkpoint inhibitors to combine with chemotherapy to boost pathological and clinical response rates. Also, future work will include exploring potential other synergies, in particular with radiation therapy. So to conclude, I would like to thank the LYSAT Lab and the Department of Surgery in Trinity St. James's Cancer Institute, in particular the clinical teams in St. James's Hospital, without which this research wouldn't have been carried out. Thank you, and I'm happy to take any questions.
Well, Maria, thank you for putting across a really complex series of investigations in such a clear manner. Can I ask Derek Alderson to take it up?
Congratulations, that's an enormous amount of work. You said, though, that there was a correlation between the chemotherapy effect, as it were, downstaging, and the immunomodulation that you've seen. Now, a correlation is one thing, and something that's biologically meaningful could be something completely different. So could you give us some idea of how strong that correlation is?
Yeah, that's a great question. So in terms of the correlations, there was about 14 to 15 patients included in this study. We carried out the Spearson correlation, and we found that there was strong positive correlations with the CTLA-4 expression and tumor regression rate. In terms of how these chemotherapies downregulated and altered CD69 and CD27 expression in patients, we saw that this happened in vivo, but we also took immune cells and treated them ex vivo and treated with chemotherapy, and we saw the same effect. So based on that, we could conclude that these chemotherapies were directly altering T cell activation status.
I wasn't quite sure what the relative timing was of your measurement of your immunological markers related to the timing of the chemotherapy, because these patients are usually going to receive multiple cycles, and how they responded both clinically and oncologically will determine just how many cycles they got. So,
yeah, exactly. That's a great question. So these post-treatment tumor samples, they were taken at the time of surgery. So not all of the patients, it's usually, so some of them would have received the 4 cycles of neoadjuvant FLOT, whereas others may not have. And also it would have been 6 weeks post-chemotherapy treatment that we would have screened these markers in vivo. However, We did take PBMCs from the blood of these patients without any treatment, and we did treat directly with chemotherapy for 48 hours. And we saw that the same downregulation or upregulation in T cell activation markers as well.
Okay, thank you.
John Alberti.
Thank you, Dr. Davern. Congratulations, beautifully presented. I commend you for your fluency in this field because it is quite difficult, and you took a difficult topic and made it understandable. And I almost wanted to take like a handful of Ritalin before you started because you only had 7 minutes to present this very difficult thing. Anyway.
Your question's longer than our talk.
Yeah, it is. No, you're supposed to say, is there a question there? Yeah, that's right. So I want to know, maybe this is the same question, to what extent were these findings a product of experiment itself versus clinically meaningful? In other words, the tumor microenvironment we're learning is very important, obviously. And, you've done a lot of these ex vivo. And so how do you account for the lack of stroma, the lack of macrophages, the lack of other tumor cell lines that could influence this outcome? Now, I understand, your aims were very clear, that you wanted to study one thing, and you did it very well. But can you just speak to the tumor microenvironment loss and the readouts as a function of experiment, not of what's happening in vivo? Thank you.
Yeah, that's actually an excellent question. And it's something that we considered as well. So I didn't have time to present all of our data on this, but we did gather tumor biopsy conditioned media. So we generated that by using the biopsies where we cultured them in vitro for 24 hours. And then we harvested that tumor biopsy conditioned media, and we treated treatment naive PBMCs with this tumor biopsy conditioned media. And we still saw an increase in CD69 and downregulation of CD27 with either the post-flot or post-cross tumor biopsy conditioned media. On top of that, the tumor biopsies, these were taken post-treatment. So we processed these immediately at the time of surgery. We'd still see the effects of the tumor microenvironment at that point, because we would look at the checkpoints or the activation markers immediately. In terms of the PBMCs then, where we treated with chemotherapy ex vivo, they were really examining the direct effects of chemotherapy on the immune cells. But whereby we use the tumor conditioned media and screen the tumor biopsies straight away after they were taken, that would help include the effects of the tumor microenvironment as well.
Well done, Maria. And last questions from Dr. Kibbe.
I will be brief. One question. Very well done, by the way. Very well done. You showed this slide where you evaluated several of the checkpoint inhibitors, and there were many of them that actually increased. So I'm just curious, how did you study the two that you showed? Why did you decide to do the two that you showed us? Or are you planning on looking at every single one, and these are just the first two? Because there may be some that are better than others.
So we did look at all of the immune checkpoints as well. So interestingly, we found that when you treated PBMCs ex vivo with chemotherapy treatment, we saw an increase in several of these inhibitory immune checkpoints on T cells. We found that TIM3 and CTLA4 were actually downregulated on these T cells when we treated with chemotherapy ex vivo on the PBMCs. When we looked at the biopsies post-treatment, we didn't see a significant alteration in most of the immune checkpoints. However, these patients would have gotten chemotherapy for about 6 weeks at this point, but we did still see the downregulation in TIM3 and LAG3. So I think the timing of the chemotherapies do have a substantial impact on the expression of these markers as well.
Thank you.
Well, Maria, thank you so much, and congratulations. And I'm afraid our jury has got a difficult task, but what is reassuring to me is that these presentations could be heard at any surgical forum in the world, and we would have been proud of them. So well done.